Okay, it looks like Fernanda's going to be running just a tad bit late, so it's my privilege to introduce our first speaker today. Doug completed his bachelor's degree in biological sciences at the Federal University of Rio Grande do Sul in Brazil, where he started his career researching stressful memories and endocannabinoid system. During his PhD training at Federal University of Sao Paulo, he studied the role that the memory generalization of animal models for addiction. Currently, he is a postdoctoral research fellow in the lab of Dr. Fabrizio Del Monte, and that's at UT Health. In his postdoc, he is investigating the neural pathways and mechanisms for controlling the balance between innate fear and reward seeking. Currently, he is in the process of transitioning from postdoc to faculty and wants to focus his lab on neural correlates and circuits of sugar addiction. So Doug, we'll turn it over to you.
All right. Can you guys hear me?
Yes.
All right, great. Let me share my screen. Good. Can you see my screen?
Yes, looks good.
Good. All right, thanks a lot, Suzanne, for the introduction. I'm very happy to be here. I appreciate the invitation. And today I'm going to share one of my postdoc work in which we describe a hypothalamic thalamus striatal circuit that controls the approach and avoidance conflict in rats. As you said, I'm a postdoc in Dr. Fabrizio Del Monte lab, and I started together with the lab, so it's been a very exciting journal, and I'm very excited to share this story with you guys. I want to start talking about fear disorders. So it's a very common problem in the US. About 30% of the population has some sort of fear disorder. It's very cost around $40 billion per year, and the recurrence it's very high. So the treatment it's complicated. About 40% of the people don't respond to the conventional treatment. And it's a kind of disorder that has a lot of comorbidity. One of these is eating disorders. So fear-associated disorder present high comorbidity with eating disorders, such as bulimic patients. It's known that they suffer for fear-related disorder, and it's also related with obesity. So it increases the rate for 1 times higher if people has any kind of fear disorder, such as PTSD. As a biologist, I always ask myself, how come some mental disorders and emotions can be integrated? So we have to think, how does that integrate in nature? And we have to think about the integration of these both situations, these both disorders. Think about foraging. So foraging is a well-preserved behavior in nature. Animals can forage for food, for shelter, for water, and it's widespread in the animal kingdom. And foraging behavior is not a neutral behavior. It involves some risk. So it's a very common problem in nature, whether it's safe to search for food, but at the same time, avoid predation. So although this is a problem that most of the animals face in nature, we still don't know which circuits of the brain are computing the balance between food seeking and avoid potential threats. And we think that understanding this circuit can be a cue and can trigger some ideas to understand how this comorbid disease are integrated as well. So this is the main question of our project, is to investigate the neural circuits that are integrating food-related information with threat. To address this question, we initially developed a ethologically relevant model in which animals are initially trained to press a lever and retrieve sucrose pellets during audiovisual cues. As you can appreciate here, can you guys see my mouse? Suzanne?
Yes.
Yes,
yes,
good.
we can see it.
So as you can appreciate here, the animals discriminate pretty quick between the cue onset from the cue offset. As soon as the animals learn to self-administer sucrose, we transfer them to a different context. In this context, it's a little bit different. It's a larger arena, and we have what we call this food area. We have the center, and we have the hidden area. And the food area is the same place in which we're going to introduce a conflict. This conflict is induced by the cat saliva. So we're going to place here in the odor compartment the cat saliva, and the animals will need to overcome the fear of the predator if they want to search for sucrose. I have a short video clip to show you how the behavior of the animals looks like. So initially the animals are experiencing the sucrose retrieval in a neutral odor. So there is no threat here. This is a normal behavior of the animal looking for food. However, the same animal the following day, when we introduce the cat odor here, we see a completely different behavior. So the animals, they approach, they risk assess, they're unsure about going or not going, and they ended up deciding staying hide in this hidden area. When we quantify these behaviors, we can see that there is an increase in the defensive responses, quantified by the avoidance, freezing, and risk assessment. When you look at the locomotor activity of these animals is also reduced. The cat odor group explore less the box, whereas the neutral odor group spend most of the time exploring the food compartment, as you can appreciate here on the top as well for the heat map. So the cat odor group spend most of the time here in the hidden area, whereas the neutral odor explore a lot the food compartment. So the cat odor was also able to suppress, as you could see in the video, the food-seeking behavior, reducing the approach time and also pretty much abolishing the number of presses per minute of the animals. So we ask which areas of the brain are being activated by the predator odor that seems to induce such a strong defensive response. So we expose the animals to the cat saliva and quantify the CFOS. CFOS is a neuronal activity marker, classically described in the literature in many areas of the brain. And we replicate in our model here, showing that the medial amygdala, the ventromedial hypothalamus, and the prelimbic area are also activated by the cat saliva. Interestingly, we found that this portion of the thalamus, the anterior portion of the thalamus, is also being recruited by the cat odor. The paraventricular nucleus of the thalamus, which I call for now PVT, it's a structure of the thalamus that sends and receives projections to areas related with stress and fear, and also food-seeking response. So this gives us the idea that PVT could be a potential structure to be integrating these both behaviors. In order to address this point, we can record the neurons in the PVT, placing an electrode in the anterior PVT, and now exposing the animals to the conflict task. The difference here in the recording settings from the video that I showed you is that I'm going to combine both moments, in the neutral odor, food seeking only, with the conflict in the same day. So basically the animals will have a food seeking phase, a cat odor phase, and a conflict phase. Therefore, we can track the cells across the different phases and track the same cells in the session. When we look to the spontaneous activity chains after each one of the phases, we saw a group of neurons that changed for the food seeking phase, another group of neurons that changed for the cat odor, another set of neurons that changed for the conflict phase only, and a big group of non-selective neurons. This gives us the idea that PVT is indeed encoding the integration of all these situations. When you take a closer look on the excitatory responses of these neurons, only on the onset of the food cues, we observe a very interesting pattern here. So we see excitation for some neurons when the food cues are presented. And interestingly, the same cell in the presence of the cat odor doesn't show the same pattern. So the cat odor during the conflict abolished the excitatory response of these neurons in the PVT. The same is true for this quick inhibition that we see here, this 300 milliseconds inhibition induced by the food cue. It's abolished when the conflict is present. So we ask whether the APVT is necessary to mediate the approach avoidance conflict. So here we did a classic pharmacological inhibition experiment. So we inhibit the PVT using mucimol, which is a GABA-A agonist, so we can silence the structure during the conflict. And we see there is a reduction in the avoidance time and an increase in the approach time. Interestingly, this inhibition of PVT has no effect when we present the animals to the cat odor alone or to the food cues alone, suggesting that PVT is indeed important for the integration of both stimuli. Now we want to understand which efferents of PVT are controlling the food seeking and the predator trap. So we know that the PVT sends dense projections to the nucleus accumbens, which is being implicated with reward and motivation. So we ask whether the anterior PVT neurons that project to the nucleus accumbens are regulating the food seeking during the conflict. To do that, we use two different viral vectors doing what we call intersectional approach. We inject a retrograde virus in the nucleus accumbens that travels back to the soma of the neurons, and then we inject the inhibitory dreads in the PVT. The dreads is a technique that's widespread in the literature, and it's used to silence or activate structures of the brain. We can put an artificial receptor that is activated by an artificial ligand, and in this case we can inhibit this pathway. So basically, with this approach we can silence the neurons from PVT to nucleus accumbens during the conflict. When we silence this pathway, we see there is a reduction in the defensive response and an increase in the food-seeking response of the rats. So from now we learn that the PVT neurons are indeed integrating both behaviors, and this specific pathway is also important for the conflict. But we haven't analyzed any signal, any molecular signature, specific molecular signature. So we ask whether there is a subpopulation of PVT that might be involved in this conflict. When you look at the literature, we learn that CRF neurons, the corticotrophin releasing factor, are present in PVT. And we know that these neurons are important for stress. So we ask whether the APVT CRF neurons are being activated by the predator odor. So we label these CRF neurons with a combination of two viruses, one that is specific for CRF neurons and another one that is a reporter, expose the animals to the cat saliva, and quantify the neuronal activity with CFOS. We see that indeed CRF neurons are being recruited by the cat odor. So now we ask whether the PVT CRF neurons are also necessary to regulate the food seeking during the conflict task. To do that, we use the inhibitory dreads again, but now the difference here, we're going to label exclusively the CRF neurons in the PVT, and we're going to inhibit these neurons during the conflict. When we inhibit the CRF neurons in the PVT, there is a reduction in the defensive response and an increase in the food-seeking response, suggesting that the APVT CRF neurons are indeed recruited during the conflict. When we record the CRF neurons specifically during the task, and we can do that combining the single unit recording with optogenetics, and then in the end of the session we shine a light in these neurons that were tagged with channel rhodopsin, and therefore the neurons that respond to the laser are CRF neurons. When we label and record the CRF neurons, we saw a group of neurons that are activated during the conflict, and we looked back to the food-seeking phase. Those neurons were not responsive. The same is true for the inhibitory responses observed in the CRF neurons. When we look back to these neurons during the food-seeking phase, these neurons were not being recruited. So this means that the CRF neurons are being recruited for conflicting situations. And now we want to see where are these anatomical targets. What are the anatomical targets of the CRF neurons that are deriving from PVT? So we label these neurons again in the PVT, and we look for the projections of these neurons. So we saw dense projections to many areas of the brain related with reward and fear, such as central amygdala, subiculum, bad nucleus, hypothalamus, and I would like to highlight here dense projections to the nucleus accumbens. So we ask it now, if this projection from APVT, the CRF neurons that projects to the nucleus accumbens, are playing a role in the food seeking. To do that, we did a different approach now. We inject a combination of the CRF specific virus with channel rhodopsin. We place the optical fiber on the nucleus accumbens in order to activate these neurons. The idea here is try to recapitulate the effect of the cat odor over the food seeking. And now here the conflict is artificial. So we're going to illuminate this circuit that it seems to be an aversive circuit to see what happened with the food seeking behavior. So we observe there is a reduction in the presses per minute and an increase in the latency of the animals to press. Indeed, this circuit seems to suppress the food seeking when it's activated. I have a video clip to show you. So here in the left panel, laser off period, and here's the laser on. So same animal in different moments, behaving completely different. So the lasers suppress the food-seeking activity. When we look at another task, which is called real-time place preference, and we illuminate one side of the box, this is what we call the laser on side, and compare the behavior of the animals with the laser off, we see that the animals spend most of the time in the laser off period, and they walk much less in the size of the laser, meaning that this stimulation is aversive. So now we ask from where the predator odor information is arriving in the PVT. We know that the PVT doesn't receive direct inputs from the olfactory bulb. So we ask where this olfactory information is arriving in the PVT. So now we did what we call monosynaptic rabies virus tracing to see which areas of the brain are projecting to the CRF neurons. And one area that we observe that is projecting to the CRF neurons in the PVT is the ventromedial hypothalamus. The ventromedial hypothalamus, it's implicated classically in the literature with predator recognition. And these neurons are indeed projecting to the CRF neurons, suggesting that might be a potential candidate to be the source of the predator information. Interestingly, when we label the projections from VMH to the PVT and expose the animals to the predator odor, we see that indeed this pathway, VMH to PVT, is being recruited when we expose the animals to the predator odor and co-label this pathway with CFOS. We see that this pathway is being recruited by the detection of the predator odor. So we thought that the VMH neurons convey the predator-related information to the PVT during the conflict. To test that, we did exactly the same intersectional approach, but now we want to label the VMH neurons that projects to the PVT, and we want to silence them using the DRADs, inhibitory DRADs. When we silence the VMH to PVT pathway, we see a reduction in the defensive behavior. But interestingly, no difference was found in the food seeking, suggesting that indeed this pathway, VMH to PVT, is the pathway that is sending the predator information to the CRF neurons. So to summarize and propose a model of our data on the light of the literature, we know that the predator odor activates the olfactory bulb, that's accessory olfactory bulb. This accessory olfactory bulb sends downstream projections to this classic pathway, medial amygdala, ventromedial hypothalamus, and PAG, inducing defensive response. This was previously described in the literature and we replicate in our model. We highlight here the APVT, especially the CRF positive cells, in which we think that in a normal situation in which animals are foraging for food, the food cues are inhibiting the CRF neurons, and therefore the animals are pressing for food and foraging for food normally. However, when the cat is present, when there is a threat there, the VMH neurons are activated and sending information to the CRF neurons. Therefore, these CRF neurons are more activated, sending activation downstream to the nucleus accumbens, suppressing the food-seeking response. So with that, I would like to appreciate and say thank you everybody in the Dumont lab, especially Zhu Zhang, that was fundamental during this project. He's a grad student in our lab. Of course, Dr. Dumonte for being a fantastic mentor and for giving me the opportunity to learn a lot during these years in this postdoc. Our collaborators, Dr. Deepe Lee, Dr. Kirwag, Dr. Barling, Dr. Justice, and of course, the funding source of our lab. I will be very happy to clarify and ask any and respond any question and also receive any recommendations. Thank you.
Thank you, Doug. It was really amazing. I have to say that I've been familiar with your work, but listening to the whole story again, it's really amazing. I'm learning always one small piece at a time. I have a question in general about this mechanism. So do you think that other type of rewards, like for instance, in the case of substance abuse disorder, would play the same role, or would they be controlled by the same pathways that for food?
Yeah, that's a great question. There is indeed data in the literature with opioids suggesting that the PVT to accumbens pathway, it's an important pathway for withdrawal feelings. So yes, I think drugs can indeed recruit the same pathway, especially PVT to accumbens. And we also know that PVT has opioid receptors. I'm not sure about like cocaine. I know alcohol also recruits PVT. So there's a lot of literature on the paraventricular thalamus and substance use that are describing the important role of the PVT and its circuits in substance use disorders as well. We also have projects that we're running in the lab trying to investigate upstream connections like PL to PVT and opioids. So this is a project of Kana. Here's a picture. He's a grad student in the lab. So he's investigating the role of PL to PVT and also PVT itself in an opioid project. But this specific pathway from PVT to accumbens, we still don't know in our hands. But we know there is this science paper proposing that the pathway PVT accumbens is important for the withdrawal syndrome.
And what about relapse? Do you think that would also play a role?
Yeah, I don't remember off the top of my head, but I think if I'm not wrong, they did this experiment on relapse. And if I'm not wrong, if I'm not mistaken, it seems that it's really related with the withdrawal syndrome. Although they did it, the naloxone model, right? So I'm not sure about like a more natural withdrawal syndrome if you expose the animal just to a withdrawal period without inducing with naloxone, if that would recruit. But I think if I'm not wrong, the relapse was not recruited for this specific pathway PVT to accumbens. I can check better and came back with you.
Well, thank you so much. Any other questions?
Doug, this is Samina Salim from University of Houston. I just had a very nice talk, very interesting. Just a quick technical question. So did you spray the cat odor? How did that work?
No, we collect the cat saliva here as supplementary slide here. So this is the way we collect the saliva. So we have a partnership with a clinic that they do spay and neuter. And then when the animals are recovering from the surgery, I pretty much come over with a filter paper and rub their mouth, the tongue, the salivary glands to collect the saliva. And then the saliva is impregnated in a filter paper. And then we have to wet, because the source of the saliva, it's a peptide. The source of the fear source, let's put it this way, is a peptide. So you need to wet the filter paper, and then you have like a mesh, as you could see in the video. So there's no way to spray it. Although there is some evidence that there might be some volatile compounds, the main source that was described in the literature of the fearful feeling, it's a peptide that needs the vehicle, in this case water, just water that you wet the filter paper, and when the animals touch and lick, they also lick during the investigation period. I didn't show in the video, but there is a moment in which they detect the cat saliva that involves a lot of investigation like nose touching and licking. and therefore you can access the accessory olfactory bulb with this peptide, triggering the response.
I see. Thank you.
Thank you. We still have a minute or two for questions. Any more questions? Okay, so if there are no further questions, I'd like to thank Dr. Engley for the great presentation, and we can move on to the next speaker.
Thank you.
Thank you, everyone. Okay. Okay, so our next speaker is Dr. Brian Denny. Dr. Brian Denny is an assistant professor of psychological sciences at Rice University, and he's the director of the Translational Social Cognitive and Affective Neuroscience Lab. He received his bachelor degree in psychology in 2005 from Stanford University and his PhD in psychology in 2012 from Columbia University. He completed postdoctoral training in translational applications of social cognitive neuroscience at the Icahn School of Medicine at Mount Sinai before coming to Rice in 2016. In 2018, he was named a Rising Star by the Association for Psychological Science. So today, we're happy to host his presentation entitled Examining the Efficacy and Mechanism of Emotion Regulation Training, Behavioral, Linguistic, and Neural Evidence. Thank you, and it's your time, Dr. Denny.
Thank you so much. Very much appreciated, Fernanda. And everyone, can you see my screen and hear me okay?
We can, both.
Perfect. Yeah. Well, thank you so much for the invitation. Really appreciate it, and happy to talk to you today about emotion regulation. And emotion regulation, particularly in the context of mental health, is kind of an omnipresent construct. So within psychology, emotion regulation is really connected to every other domain and serves as a very useful pathway to try to understand emotional experience and whether we can improve in our ability to adaptively respond to stimuli that we encounter, including negative stimuli that we encounter. Because try as we might to avoid it, we can't. Interpersonal conflict, threatening stimuli that we may face, difficulties at work, or even something silly like getting into a fender bender. So if we just pause on the idea that got into a fender bender, you take one event like that, and responses can really vary. You can kind of, just despair. You may engage in more maladaptive health behaviors as a result of it, or you may kind of take it all in stride and be okay with it. And so from this one emotional event, if 100 people experience it, you may get not one response, but 100 different types of responses, right? From one event. So this is related to the model of emotion regulation that I'm going to talk to you about today. You have a stimulus in context, you have a situation that you're experiencing to which you devote attention. And crucially, you make a judgment, an appraisal judgment about what you're attending to. And so it's this process of appraisal that I'm going to be focusing on today, particularly, in the sense that it's not so much what happens, but it's how we interpret what happens that's very important in determining our emotional responses. So I'm going to talk to you about how emotions are regulated in the context of this model that's been very influential from James Gross. And in the example of the car fender bender, you could first modify your the situation by, for example, leaving the parking lot and avoiding the situation entirely and all thoughts about it, you could modulate your attention that you devote to that situation by distracting yourself and focusing on unrelated things. You could modulate your response by suppressing and trying to show a poker face, or you could cognitively change how you think about what you experienced and emphasize that no one was hurt and realize that it's only a material thing. So the question I'm going to focus on in a few studies that I'm going to present to you is whether we can improve our ability to regulate our emotions over time. So is it possible that in repeatedly practicing ways of regulating emotion that health and emotional experience can be improved? So challenging events are all around us, but can we improve our ability to regulate our emotions over time? And in the clinical domain, we have contact with this question, of course, in that we know that modalities, psychotherapy modalities like CBT are effective, but crucially, they're often lengthy, involving many months of treatment. And crucially, in something that I'm going to unpack, many strategies are trained, so it's quite heterogeneous. So a variety of cognitively and behaviorally focused strategies are trained. So this is true in CBT as well as DBT. And in healthy adults, there's even less evidence in direct contact with the question of whether we can improve our ability to regulate our emotions over time with emotion regulation training. So the overall questions I'm going to discuss, challenging events are all around us, but can we improve our ability to regulate our emotions over time? This is the whether and the when question. And I also briefly want to touch on a study that's formative to what I'm going to finish with, what neural mechanisms drive training effects. And then I'll finish up with recent work with a graduate student in my lab, Anushka Shahane, with whether we can predict emotional regulation efficacy via novel computational algorithms to analyze natural language use. So both questions 2 and 3 are how questions or mechanistic questions seeking to understand emotion regulation training further. So for the first study that I'll describe to you that sets the table, in a way, imagine that you're a mail carrier, and as a mail carrier, you encounter, unfortunately, a particularly nasty dog, and you don't just encounter that dog once, you encounter that dog repeatedly, and not just the one type of dog, but many types. So negative stimuli that you're encountering of a diverse nature. And the questions are, can we get better over time if we attempt to regulate our emotions with training, and are some strategies more effective than others? In the second study that I'll talk to you about, still imagining that you're a mail carrier, after you have had an opportunity to regulate your emotions multiple times, if there's a delay of time and you don't encounter the stimuli to which you had previously regulated your emotions, and then you re-encounter a stimulus later, do the effects of having regulated your emotions persist? And if so, how? And does repeated practice matter? So in this second study will involve fMRI evidence, whereas the first one will be self-reported negative affect and questionnaire reports. And the regulation strategy that I'm going to talk about throughout the talk is cognitive reappraisal, which involves a cognitive change strategy to change the meaning of an emotional stimulus in a way that alters its emotional impact. So I'm going to be focusing on downregulation of negative emotion throughout the talk. And there are 2 reappraisal tactics that I'm going to unpack for you. Distancing versus reinterpretation. So in reappraisal by distancing, you can adopt the view of an impartial objective observer, or imagine that events are physically or temporally distant. And this is something that particularly in the last study I'm going to talk to you about that we'll explore further. Alternatively, you can reappraise by reinterpreting, by imagining a better outcome, or imagine that things are not as bad as they first seemed. So in the example of this photo above, to take the perspective of an objective impartial observer, you can say that the one woman is gesturing to the other with her palms extended and stretched outward. But in reappraisal by reinterpretation, you can say that this is only a momentary argument and they're soon going to make up and hug. So this is a general task overview for all the studies. So images are preceded by cues that indicate whether to look and respond naturally or to regulate negative emotion for the upcoming negative or neutral picture. Then there is a negative affect judgment made trial by trial. And so there are 3 task conditions: look paired with neutral, look paired with negative, and reappraise paired with negative. So in the first study, we're asking the question, can we get better over time, and are some strategies more effective than others? This is a study in healthy adults with DVs of self-reported negative affect and questionnaire reports. And so the idea was, if you encounter negative stimuli over the course of 4 sessions, and you're repeatedly regulating your emotional responses to those stimuli using reappraisal, can you improve in your ability to regulate over time as a function of whether you were trained in distancing or in reinterpretation? So at each session, participants completed the perceived stress scale, did walkthrough training with an experimenter, and then did the image-based task I described. The perceived stress scale, for those of you who aren't familiar, asks questions like, in the past few days, how often have you been upset because of something that happened unexpectedly? Or in the past few days, how often have you felt nervous and stressed? So there was a group randomly assigned to receive training in distancing, another group receiving training in reinterpretation, and another group that did not receive emotion regulation training, and it was a look-only natural history control. The images were counterbalanced across sessions and conditions with 33 participants per condition. And so here are the results on self-reported negative affect. Not much happening longitudinally for look-neutral trials. For look-negative trials, there was longitudinal reduction in the distancing training group and in the reinterpretation training group, but in distancing, the drop was greater than the no regulation group, suggesting that the reappraisal training became more natural over time to apply regulation even to images where that was not cued. And in terms of the reappraised negative trials, there were longitudinal reductions for both the distancing group and the reinterpretation group, with negative affect lower overall in the distancing group. But what was quite interesting was the perceived stress results. So starting out not significantly different from one another, the distancing group showed a longitudinal reduction over time in perceived stress that was unique vis-a-vis the other groups. So an interim summary involving study 1, can we get better over time with training? Both distancing and reinterpretation reduced task-related negative affect. But with regard to whether some strategies are more effective than others, distancing significantly reduced negative affect even in unregulated trials. And only distancing involved significant drops in perceived stress. So we interpreted this to mean that distancing training may involve a change in a stimulus-independent mindset, as opposed to reinterpretation, which is very stimulus-linked. So this motivated a follow-up study that involved fMRI, specifically focusing on distancing training. So again, imagine that you're a mail carrier and that you repeatedly encounter a negative stimulus like this dog, and it provides an opportunity for you to regulate your emotions repeatedly. Then you are switched onto a different route and time passes, but then lo and behold, you're switched back onto your original route and re-encounter the negative stimulus to which you had regulated your emotions originally. And the questions here were, do the effects of having regulated your emotions previously persist? And if so, through which neural mechanisms? And does repeated practice matter? Does it matter whether you have engaged this emotion regulation repeatedly as opposed to just one time? So this study was done in healthy adults with self-reported negative affect and fMRI as DVs. And just to orient you, for those who may not be familiar with regard to the neural bases of appraisal and reappraisal, so in a quantitative meta-analysis of 48 studies, Buell and colleagues showed that the amygdala was the only brain region consistently engaged in bottom-up appraisal, so negative emotion reactivity. But there were, so the only consistent target of regulation, but in terms of implementing that regulation, in top-down reappraisal, there was a suite of cognitive control regions that were consistently engaged by cognitive reappraisal, including the bilateral ventrolateral prefrontal cortex and bilateral dorsolateral prefrontal cortex. So this is an overview of the task paradigm in study 2, which you'll notice is very similar to what I originally presented before. And in this study, the reappraisal tactic was always distancing. So here was the design. So on the first day, you are repeatedly encountering negative stimuli, either with the instruction to reappraise or to look. But this is a massed practice emotion regulation training session with 3 presentations for reappraise and 3 for look. So it occurred outside of the scanner. Day 2, we call active regulation, was scanned with the same reappraisal task with those images that had been repeatedly reappraised, as well as images that had been repeatedly looked at, as well as new reappraised negative images that were new at active regulation, as well as new look negative images, as well as look neutral images. And then a week later, after you had, if you're the mail carrier, you'd been switched onto a new route. And then a week later, you're switched back and you have long-term re-exposure. And in the scanner, you passively view all of those trial types that you saw before, as well as novel negative images. And all of the images were counterbalanced with regard to their conditions across participants. So I'll show you data here that's been published from 17 healthy adults. And so focusing on the amygdala activity at active regulation and long-term re-exposure. At active regulation, there were 2 main effects, but no interaction. So a main effect of regulating with reappraisal versus looking and responding naturally, as well as a main effect of repeated presentation versus single presentation, but no interaction, although in a direction that we would expect. But the question is, what were the responses during long-term re-exposure? And here we did observe an interaction where responses were maintained as having been attenuated, particularly and uniquely among negative images for the repeated reappraised negative case shown here. So there was a neutralizing effect for stimuli, particularly when they had been repeatedly reappraised. So then the question became, so what sort of mechanisms in the prefrontal cortex may subserve that long-term maintenance of emotion regulation? So we examined activity in the left ventrolateral prefrontal cortex that during active regulation showed a mirrored pattern from amygdala at active regulation, here showing, again, 2 main effects and no interaction of regulation as well as repetition during active regulation. But then at long-term re-exposure, there was no difference between the repeated reappraised negative and the repeated look negative, which we interpreted to take as a retrieval effect. And we looked for other places in the prefrontal cortex that might have shown an effect greatest for repeated reappraised negative trials. and also looked for patterns of functional connectivity between the prefrontal cortex and amygdala, and did not find it. So we interpreted this to mean that the repeated reappraisal was associated with a change in the bottom-up reactivity to the stimuli at long-term reexposure, rather than being heavily mediated by prefrontal mechanisms at long-term reexposure. So we found evidence that there was long-term maintenance of the emotion regulation as reflected by the brain activity. So do effects of regulation persist over time? Yes, so there was evidence of having gotten over it, and repeated practice appeared to matter in that the effects were specific to having repeatedly reappraised a negative stimulus. And so this then leads to thinking about other mechanisms through which this happens. So neural mechanisms, but also linguistic mechanisms. And so this is work in this last study that I'm going to share with you that's recently been published with my graduate student, Anushka Shahane. And so here we focus on language-based mechanisms for psychological distancing, like the psychological distancing that I talked to you about before. And so in a series of studies with Anushka, we are actually disambiguating whether or not that distancing is occurring via objectivity, which was that first sub-tactic for distancing that I mentioned before, versus spatial or temporal distance. So imagining and increasing the perceived distance spatially of something from you, or the perceived distance temporally of something from you. And so looking at both of these sub-tactics of distancing and how they're represented via language. So there is an existing methodology to be able to quantify psychological distancing from language using something that we are calling the linguistic distancing composite metric, which leverages LINQ, linguistic inquiry and word count from James Pennebaker, that involves an assessment of first-person singular pronouns, present tense verbs, discrepancy words, articles, and words greater than 6 letters. And previous research has shown that greater linguistic distancing via the linguistic distancing composite metric is positively related to emotion regulation efficacy. But what we were interested to do was to leverage machine learning methods to generate novel classifiers, novel algorithms, to assess the extent to which a given natural language pattern is reflective of spatial temporal distancing or objective distancing. So Anushka and I have previously looked at this using the linguistic distancing composite metric and a group that had been trained in objective distancing, as well as a group that had been trained in spatial and temporal distancing. And we found that the objective group showed more associations, adaptive associations between increasing psychological distancing as assessed via the LDCM and adaptive affective and health outcomes. And fewer associations were observed for a group of healthy individuals that were trained using spatial or temporal distancing. So that motivated us to use a machine learning methodology, BERT, so bidirectional encoder representations from transformers, to be able to generate novel algorithms to assess linguistic distancing. So to our knowledge, this is the first time that something like BERT has been leveraged for linguistic analysis of distancing. And so BERT was developed by individuals at Google to use a neural network architecture that's bidirectional, which means that to assess the value of a word in a sentence in natural language, you look at both the words that precede it as well as the words that follow it, and it's a contextual, deep learning, contextual analysis machine learning method. And so what we did was we had 142 participants read and rate sentences based on objectivity and spatial temporal distance to use as annotations for this machine learning. And then I'm happy to talk about this in more detail if people are interested, but we did BERT semi-supervised training first with general sentence banks, and then we leveraged these participant ratings that we received from 142 healthy adults, and then performed BERT supervised training to be able to take the sentence ratings as annotations to feed into our BERT model to then generate algorithms that could provide a score for spatial and temporal distancing on the one hand and objective distancing on the other hand. And so we've recently published the results of this, showing that we were able to do this using a five-fold cross-validation and generate an objective algorithm as well as a spatial temporal distance algorithm, both of which were valid. And then we wanted to further test those algorithms to see if they predicted reappraisal success longitudinally. So we did this by both having different groups that were manipulated to either engage in objective distancing versus spatial temporal distancing, and then we leveraged these novel algorithms that were created for objective versus spatial temporal distancing. And so there were 39 participants in an objective group that were asked to write about pictures using impartial and objective words, and 38 participants in a faraway group that were asked to write about pictures using words that make the image seem physically and temporally far away from you. And so participants did this over the course of 5 days.
2 minutes, Brian.
Thank you. Where they used the same paradigm that we talked about before, but this time having an expressive writing window when the image was presented for 30 seconds where they could expressively write in any of the conditions, look negative, change negative, and look neutral. And so what we found was that within the objective group, increases in the regulation efficacy for the objective algorithm from T1 to T5 significantly positively predicted increases in reappraisal success from T1 to T5. We found the same thing using the FAR algorithm, but there was no significant association between change in the linguistic distancing composite metric regulation efficacy and change in reappraisal success, although it was the same pattern. So what this is showing is that we were able to validate the algorithms that we generated in showing that as people are engaging vis-a-vis the algorithms in more objective distancing over time, that's associated with greater reappraisal success, defined as greater diminishment in negative affect, in self-reported negative affect during the task. So in these unobtrusively measured analysis of natural language patterns, we're seeing that there's an association between that and the negative affect ratings that people are making during the task. Within the FAR group, there were no significant associations between changes in the regulation efficacy from the objective and FAR algorithms and changes in reappraisal success. So the summary here was that we developed novel computational algorithms that index the degree of objectivity and spatial temporal distance in language use. And then in the second study, in this last part, we showed that we validated that the objective and FAR algorithms predicted emotion regulation efficacy longitudinally. And this provides some additional evidence for the greater ability of objective distance relative to spatial temporal distance to predict emotion regulation efficacy. Although results in study 3B also reflect a deep conceptual similarity between distancing subcomponents, objective and spatial temporal distance, consistent with prior work using the linguistic distance composite metric. So we're now examining the objective and FAR algorithm performance in predicting affect in additional contexts, including outside of the lab, as well as in affectively disordered populations. And we're also examining emotion regulation training in bereaved spouses as well, including using linguistic analysis. And ultimately, innovating optimal interventions will require an understanding of the optimal matching of person factors, situation factors, and strategy factors, all aligning. So ultimately, we want to move toward personalized emotion regulation training, where we assess responses via language and behavior, experience and brain activity, and crucially, the interactions between them. And so with that, I'd like to thank my collaborators for the first two studies at Columbia and NYU, as well as my former postdoc, Rich Lopez, who worked on the last study, Chris Fagundes at Rice, and my lab at Rice, especially my grad student Anushka Shahane, whose work I showed you some of, as well as Daniel Pham and our funders. And thank you very much for your attention.
Thank you so much, Dr. Danny. That was really interesting. And actually, we don't have a lot of time left for questions. I have a question if you don't mind. So I'm actually a basic scientist, so working primarily up to rodent. That is what I do. So what are the potential, if any, behavioral models that one can use, for instance, for cognitive reappraisal in rodent? Is there anything that could be could have a translational value that could be applicable in rodent for testing, the circuitry or testing mechanisms in general?
I think that's so that's a good question. It's kind of the opposite direction of translation that we usually think, right? We usually think of translating rodent models to humans. But in this case, I think that the kind of back translation is a bit more difficult, right? Because assessing experience from rodents is very difficult to do in a way that we have the ability to do that in humans. And I think it's really interesting in that the kind of formative work from a behaviorist tradition kind of led to the increasing focus on cognition in humans, because in rodents the kind of stimulus response contingencies were able to be very well mapped out and continue to be with regard to the presentation of aversive stimuli, for example, to rats, and then observing characteristic freezing behaviors and other kind of negative responses that you observe, right? And then you can observe many different response contingencies related to that. But what was observed and what happened in psychology in the cognitive revolution was the observation that even with very similar inputs, you could get variegated outputs as a function of how people were interpreting the information that they were given, or interpreting the stimuli that were presented or that were in their environment. And so this kind of focus on cognition, I think, is certainly you can think about it in the context of other animals, but I think it's much more difficult to do so and much more difficult to assess relative to humans, where we have this much better window into emotional experience to try to survey that cognitive level of analysis, as well as linguistic analysis, which is unique to humans. But I think it grew out of this tradition, looking at stimulus and response contingencies in a more behaviorist tradition. And so I think it bears on kind of behavioral neuroscience in animal models with regard to stimulus and response, but less so at the cognitive level. Does that help it?
Yeah. Thank you so much.
Sure.
Thank you. So yeah, we are actually 2 minutes past the hour. And so I would like to thank again the speakers for today. And it was quite interesting, completely different domains. And yeah, thank you so much. Suzanne, anything to add?
No, just really appreciate the speakers and everybody that attended today. So thank you both and all of you attendees.
Yeah, thank you again. Bye-bye. Bye-bye.
Thank you.